Today, we will be sketching and discussing the cell membrane. The cell membrane serves as a semi-permeable barrier between the intracellular inside of the cell and the extracellular outside of the cell. The cell membrane itself is made up of something called the phospholipid bilayer. The term bilayer refers to its two layers of phospholipids, which creates an internal environment of the cell membrane that is separate from the rest of the cell.
Each phospholipid has a polar phosphate head and a non-polar lipid tail. The heads form hydrogen bonds with water, and the tails bond with each other. This way, the heads face outwards and protect the inner tails. The polar head is hydrophilic, meaning it mixes well with water, and the non-polar tail is hydrophobic, meaning it is repelled by water. Since the cell membrane has both hydrophilic and hydrophobic parts, it is called amphipathic.
Embedded in the bilayer, there are also many proteins. This is an integral protein. These proteins are elongated and hollowed out, allowing large molecules to pass through the cell membrane. Most integral proteins are called transmembrane proteins. This means that they go through both sides of the phospholipid bilayer. In contrast to the transmembrane proteins, the membrane also has peripheral proteins. Peripheral proteins are hydrophilic and are only found on the surface of the cell membrane. Peripheral proteins can serve many functions, such as anchoring the cell membrane to the cytoskeleton, acting as receptors for cell signals, and transporting molecules.
Inside the cell membrane is a type of lipid called cholesterol, which assists in regulating the membrane's fluidity. Glycoproteins are formed by the covalent bonding of oligosaccharides, which are chains of carbohydrates, to the protein molecules in the cell membrane. The carbohydrate part of the glycoproteins often protrude into the extracellular environment and assist the membrane with cell recognition, adhesion, and signaling. Glycolipids are carbohydrates that are linked to lipids by covalent bonds. They contribute to membrane stability by forming hydrogen bonds with water molecules surrounding the cell.
Cell membranes have a wide range of functions. The primary function is to form a barrier that controls the passage of materials in and out of the cell. Other functions are carried out by proteins in the membrane. One example of these functions is junctions. An example of a junction in animal cells is a gap junction. Gap junctions are channels between neighboring cells that allow the transport of materials. Other functions of the cell membrane is carried out by enzymes.
Enzymes are a type of membrane protein. They catalyze reactions in the cell membrane when substrates attach to their active sites. For example, glucose 6-phosphatase is a type of membrane-bound enzyme that can be found in the endoplasmic reticulum.
Additionally, a main function of the cell membrane is transport, or getting substances in and out of the cell. There are two different types of transport: active transport and passive transport. In active transport, particles can move from either low concentration to high concentration, or high concentration to low concentration. Molecules have to be carried in and or out of the cell by a protein pump or vesicle, and energy is required. Conversely, in passive transport, particles only move from a low concentration to a high concentration, and they do so without using energy. These molecules can either pass through the phospholipid bilayer on their own, or may need a channel protein to facilitate their transport.
Cell recognition is when cells interact with one another through molecular signals on their cell membranes. Cell-to-cell recognition is typically carried out by glycolipids and glycoproteins on the cell membrane. The carbohydrate chains on them act like identification tags. Recognition is a way for cells to communicate with each other.
In anchorage and attachment, proteins are embedded in the membrane and cells attach to the extracellular matrix, aka the ECM, or other cells. An example of this is how integral proteins are embedded in the membrane and peripheral proteins are attached to the outside.
Finally, signal transduction is the process of signaling molecules to bond to transmembrane receptors. First, an extracellular signal is sent when a hormone binds to a receptor on the surface of the cell membrane. Then, the transmembrane receptor recognizes and bonds to a single molecule. This bond causes the receptor's shape to change conformationally, and its intracellular components are activated. The receptor then makes a series of signals that activate secondary messengers and enzymes. The signal creates a cellular response, such as activation of genes for cell division or growth, changes in the cell's metabolism, alterations in the cell's cytoskeleton, or secretion of molecules. This process allows for cellular communication and regulates cellular functions, such as metabolism and cell division. However, disruptions in the signal can cause disease, like cancer or developmental disabilities.